Okay, I just want to take a few minutes to review with you the flow of genetic information, because this is something that is going to be important for you to have an understanding of as we start to dig deeper into how Cas9 works. So briefly, the flow of genetic information is the transfer of the information that is stored in the DNA, in your nuclei, in your body, in the cells in your body, to an RNA copy, what we call an RNA transcript. That then takes that information and brings it to the ribosome, and the ribosome then translates that into a peptide or protein chain. It does that by these transfer RNAs that read every 3 nucleotides, every set of 3 nucleotides, which we call a triplet codon. And when the correct transfer RNA finds that triplet codon, for which it has an anticodon, it then adds the correct amino acid to the growing peptide chain. And once it puts all of those amino acids into the chain, then that protein can then fold into the shape that it is supposed to be in. Now, there are a few things that I want you to be aware of or think about a little bit more deeply when we talk about this process. We talk about this being an RNA copy. It holds that information that is in the DNA at that place, but it's not an exact copy, right? It is what we call a complementary copy. So let's back up. So let's talk about DNA. We know that DNA is double-stranded, and we know that it is for every one of the 4 nucleotides, adenine, thymine, guanine, and cytosine, there is one of those nucleotides that will base pair with that nucleotide. So we know that adenines and thymines base pair, A's and T's, and we know that G's and C's, guanines and cytosines, base pair. Now, we're not going to talk about the specific molecular structures that cause that base pairing to happen. But they basically fit together like puzzle pieces because the molecular structures and the bonding works so perfectly for those pairs. So we just need to know that they like to be together. Sometimes errors are made and the wrong pairs are made, but most of the time you'll have that correct base pairing. And that's really important because that keeps that information safe as our cells divide and as those genes are transcribed into proteins. So it's important that we always have the correct base pairing. Now, in addition to DNA base pairing, we can also have a DNA RNA hybrid base pairing, and that's how transcription works. Once the DNA is separated and the RNA polymerase then brings in the RNA nucleotides that base pair with that stretch of DNA that is being transcribed. So you are forming basically a DNA RNA hybrid for a short period of time, and then once that is done, then that makes that transcript and the DNA can come back together in a double strand again. But this transcript is an exact copy of the DNA that is being transcribed, but it's an exact complementary copy, okay? So it's the inverse. It's going to be, for every C there will be a G, for every T there will be an A, for every A there will be a U, because in RNA there are no T's, there are U's instead of T's, but it's just the same thing. We just think about it as the same as a thymine. It base pairs with adenine. All right, so I want you to feel really comfortable about complementarity because that is going to come up over and over again in this module. Now, in addition to DNA base pairing with itself, an RNA base pairing with DNA in order to form an RNA transcript. RNA can base pair with itself, and a DNA strand can base pair with itself. It's possible if there are stretches of complementarity in one strand of RNA or DNA for that strand to flip over onto itself and bind to itself. Sometimes they call that a hairpin, if it just it flops over and binds to itself. And that is going to be really an important point. It's something that you may not have studied in school. It's kind of a high-level concept, but it's truly everywhere in biology. It's just not something that is taught a lot, but it's a huge area of research now, and I think it's going to be more and more taught at the high school level. But this is going to be a very important part of the MAPS module, and so we are going to spend some time talking about that in the next section. Okay, so in the last section we talked about how the flow of genetic information happens, how DNA is transcribed into RNA and RNA is translated into protein, and that does happen a lot in our bodies, as we know. But that isn't always the case. So all RNA is not destined to be protein, to be translated into protein. And you probably are already aware of that. You just don't maybe think of it that way. But there's a lot of RNA that is called non-coding RNA, which means it doesn't code for proteins. It is coded to be a molecular machine all by itself. It's going to have secondary and tertiary structure and act like a molecular machine, like proteins do. And you know of some of these already, okay? So if we look at this, we talk about transfer RNA. Well, transfer RNA is non-coding RNA. It's a molecular machine all by itself. We talk about the ribosome, ribosomal RNA, and that's what we have right here. Okay, it is a molecular machine made up of primarily RNA. So how does that work? So we know that proteins, based on their sequence, fold into a very specific shape that is important for them to carry out the jobs that they have, or the job that they have. And RNA is similar. It folds into these very complex shapes based on a couple of different factors. And one of them is complementarity that we were talking about before. So base pairing within itself. And if you look at a tRNA, you can see that it is just one strand of RNA that circles through and then base pairs with itself. And then what it does is it turns into a very twisted, complex 3D shape because of a stickiness of the backbone that we're not going to go into, but the fact that it's ribonucleic acid and not deoxyribonucleic acid makes it have a sticky backbone that will allow it to contort and stay into these shapes. So on this graphic you can see that that RNA, as we know, is single-stranded. And if it is destined to be a transfer RNA and not translated into a protein, it then can base pair in this kind of cloverleaf pattern, they call it, because it's got these regions of complementarity that we were talking about before. And then, because it's got that sticky backbone, it then twists into this three-dimensional shape that is very specific for its sequence. So this structure is very specific and very required so that it fits into the ribosome just right so that it can add that amino acid to the end of the growing peptide strand, right? So that is one example of a non-coding RNA. Okay, so in addition to transfer RNA, you are also aware of of something called a ribosome, and that's what this purple structure is here, but this is what it really looks more like. Okay, so the gray pieces are actually proteins that are sprinkled on it, but they're not all that critical to the function of the machine itself. The ribosome is dependent upon the structure of this RNA that has that large subunit and the small subunit that base pairs with itself and contorts into this three-dimensional structure in a very specific manner based on its sequence. And so this is an area of study now that we understand that this is just the tip of the iceberg. We've known about these a long time because they're important in proteins, in the production of proteins. And for a long time, all that anybody cared about was proteins. But it turns out that there are probably thousands of non-coding RNAs of all different types and all different jobs in our genome. And we are, researchers are finding out more and more about that all the time. So this is an area that I think you may not know a lot about now, but you will hear more about in the future, especially if you continue on in biology. And we're going to talk about it in this section because we're going to talk about another kind of non-coding RNA that is involved with Cas9 function, and it's the guide RNA that Cas9 has to bind to. So we'll talk about that later, but I just want you to appreciate the fact that not all RNA ends up as translating into protein. Only the messenger RNA does. There's lots of other RNAs, and they all have specific structures and functions all their own. Now, from here, I want you to work on an activity in the module, and it is trying to understand what happens, what these non-coding RNAs that have these regions of complementarity, how does that look in the genome? Okay, so we are going to create our own RNA structure. We're going to have a stretch of RNA with a hairpin turn, and then a complementary region after that. And they also call that a stem loop structure because it's got that loop in it. Okay, so a stem loop structure, and we're going to see more of those in this module. So you're going to create your own. Hopefully you have some foam nucleotides available to you. If not, you can make something out of paper. You can get like pipe cleaners and beads and do something that way. So try to figure things out. You can be resourceful, and you can model with just about anything. And we are going to do that later in the module. We're going to find some garbage to model our Cas9 with later on. So we're going to reduce, reuse, and recycle. So that's that. Work on the activity, and then I will come back and we'll talk about that later.